Hello, and welcome to Chapter 3, Forming a New Life. I always like to begin with a little review from last week. You were introduced to the 8 major periods of human development and to 5 perspectives on human development, along with some of the important theories. One of the theories you were introduced to was Piaget's cognitive theory. In Piaget's cognitive theory of human development, what did he call the process through which adaptation occurs? If you said assimilation and accommodation, you are correct. Let's try one more. If you are conducting a study of the effect of environmental factors on one's communication skills, and you use naturalistic observation to collect data, where are you most likely to observe participants? Yes, in the real life setting without any attempt to manipulate their behavior. If I'm going to study nurses, then I might study them in the setting in which they work, maybe a hospital or a clinic. Let's quickly go over your learning objectives for this chapter. By the end of the chapter, you should be able to explain how conception occurs and what causes multiple births. Describe the mechanisms of heredity in normal and abnormal human development. Explain how heredity and environment interact in human development. Describe prenatal development, including environmental influences. And discuss the importance of high-quality prenatal care. Conceiving new life is obviously a beautiful experience for most people. Most people think of development from the point of birth, but development starts at conception as the sperm and egg meet. At the point the sperm and the egg meet, a zygote, which is a single cell, is the new human being. It is created in that moment. Fertilization or conception is the process by which the gametes, the sperm and the ovum, combine to create a single cell called the zygote. Women have all of their eggs or ova that they will have at birth. How many do you think we have when we are born? Any guesses? It's about 2 million immature ova in the 2 ovaries. There are many false perceptions about pregnancy. As nursing students, you need to learn the facts and to use correct terminology. Let's review the process briefly. When a couple has intercourse, the sperm travel through the cervix and up the fallopian tubes to meet the ovum. Before the sperm and the ovum can meet, the ovum must have been released. When menstruation begins, it marks the beginning of a new cycle. After the uterus sheds its lining, and then in response to hormones, the lining thickens and the egg follicles mature. Around day 12 to 13, the cervical mucus is increased. Around day 14, the egg is released and only lives about 24 hours. This is when a person can become pregnant. The sperm and egg generally meet in the fallopian tubes. The fertilized egg then travels down to the uterus where it may implant. Do you know how many eggs a woman produces? She is born with all the eggs she will have, but she only releases one every 28 days. Take a look at this chart that you have. You can find many similar charts online. I would highly recommend that you find a couple of these, the ones that suit you, and that you take the time to learn the process of conception, the fertile periods, and the different hormones that interact at each time. There is also a process by which the sperm are produced. Sperm develop as tiny round cells in seminiferous tubules of the testicles. The hormones from the testicles and the pituitary gland help the sperm mature. They then travel to the epidermis and develop further for several weeks. They migrate to the vas deferens. They travel to the seminal gland where they're nourished by seminal fluid. Then they travel to the prostate gland and mixed with prostate fluids. And then they exit through the urethra during ejaculation. How many sperm does a mature male produce? Any ideas? Well, they produce several 100 million a day. Let's talk a little bit about multiple births. We know that the woman generally only produces one egg every 28 days, but we also know that a man produces hundreds of millions of sperm. So if the man is producing this huge number of sperm and the woman's only producing one egg, how do we end up with multiple births? Well, the truth is that women can sometimes release more than one egg. This is more likely as you get older. The most common multiple births are twins, but other multiple births are possible. There are two ways to have multiple births. You can have dizygotic twins, and those are fraternal twins. They're no more similar than any other two siblings. Two different ova are fertilized by two different sperm. So they're genetically just like other siblings. They tend to run in families and result from multiple eggs being released. It's possibly genetic or passed from mother to daughter. Dizygotic twin births have increased by 76% from 1980 to 2017, and it's probably due to having babies at older ages and the use of fertility drugs. The other type is monozygotic twins. Monozygotic twins are identical twins. They result from the division of a single zygote after fertilization. So one egg fertilized by one sperm. They are genetically similar but may look different due to environmental factors. This is less common with only about 4 per 1,000 live births. Multiple births are associated with increased risk of pregnancy complications, including premature delivery, low birth weight infants, and disability or death of the infants. Let's review. Which types of twins are genetically identical? Yes, monozygotic twins are genetically identical. Genetics is the study of heredity. Heredity influences everything from eye color to height to intelligence and even personality. The genetic code is transmitted by DNA, which stands for deoxyribonucleic acid. The base elements of DNA are adenine, thymine, cytosine, and guanine, and they make up the genetic code. I'm not going into this in detail since you should cover it in other classes, And you probably covered this in high school. For now, you need a basic understanding. Chromosomes are coils of DNA that consist of smaller segments called genes, which are the functional units of heredity. The complete sequence of genes in the human body constitutes the human genome. As cells divide, they differentiate, specializing in a variety of complex functions. If a mistake in copying is made, permanent alterations in genes can occur, resulting in mutations. What determines the sex is one pair of the chromosomes. At conception, the 23 chromosomes of the sperm and the 23 chromosomes of the ovum form 23 pairs for a complete set of 46 chromosomes. At conception, the zygote, which is one cell, has all its biologic information to form a unique human being. In essence, it has formed a unique human being. 22 pairs are autosomes, which are not related to sexual expression. The 23rd pair are sex chromosomes, one from the father and one from the mother. Let's talk about that a little more. Figure 3.3 in your textbook will show you what determines genetic sex. The father has an X chromosome and a Y chromosome. The mother has two X chromosomes. Boys receive an X chromosome from the mother and a Y chromosome from the father. Girls receive an X chromosome from both the mother and the father. So genetic sex for a female is XX and genetic sex for a male is XY. Initially, whether the zygote has an XX or an XY sex chromosome, the development looks alike. Exposure of a genetic male embryo to high levels of testosterone usually lead to the development of a male body with male sex organs. In the absence of hormonal influences, the sex organs will appear female. Likewise, in females, there are influences. For example, females have two X chromosomes. For a long time, we believed that one set was turned off. We now know that about 75% of one set is turned off. But if there is a defect or other event, it can be turned on. It is one of the reasons it is believed that women generally are healthier longer. I have provided you a flow diagram that provides you details of the differentiation cascade of the male genitalia system. While this is more detailed than you need for this class, I think it will be interesting, and it is information that you'll need to know in the future. We also have dominant and recessive inheritance. Some characteristics like hair color or eye color are controlled by single dominant transmission, while others are impacted by many genes working together. Alleles are genes that can produce alternative expressions of a characteristic like hair color. One allele comes from the mom and the other from the dad. When both of the alleles are the same, the person is homozygous. When both alleles are different, the person is heterozygous. In dominant inheritance, the dominant allele is always expressed, meaning it shows up as a trait in that person. Let's think about this very simply with eye color. We know that brown eyes are dominant and blue eyes are recessive. In recessive inheritance, the person must have two recessive alleles, one from each parent. If you look at the eyes on the left, you will notice that both parents had brown eyes, but each carried a blue eye recessive gene. It took both parents donating the blue eye recessive gene to have a blue-eyed baby. My father had brown eyes and brown hair. My mother had blonde hair and blue eyes. All three of my brothers have blonde hair and blue eyes. I ended up with brown hair and brown eyes. Look at the chart. What were my chances of having brown eyes? You will need to look at the chart on the right. It would be about a 50% chance. Relatively few traits are determined in this simple fashion. Most traits, however, result from polygenetic inheritance, which is the interaction of many genes. We also have genotypes and phenotypes. My brown hair is a phenotype. A phenotype is the observable characteristic of a person, eye or hair color as an example. A genotype is the genetic makeup of the person containing both expressed and unexpressed characteristics. The dominant and recessive genes you can't see from the prior slide. Multifactorial transmission is a combination of genetics and environmental factors, nature and nurture. Intelligence is a common example. The environment can trigger some genes to turn on or off. This is referred to as epigenesis. Epigenetic changes can occur throughout life in response to environmental factors such as nutrition, smoking, sleep habits, stress, and physical activity, contributing to certain ailments such as cancer, diabetes, and heart disease. Genetic imprinting is also important for you to understand. One example of epigenetics is genetic imprinting, which is the differential expression of certain genetic traits depending on whether the trait is inherited by the mother or the father, or inherited from the mother or the father. Problems in genome imprinting may explain why a child with an asthmatic mother is more likely to develop asthma than a child with an asthmatic father. When imprinting is disrupted, abnormal fetal growth or congenital growth disorders may result. Genetic and chromosomal abnormalities are not uncommon. About 21% of all deaths in the first year of life are due to birth disorders, yet only 3% of babies have a birth disorder. The most common of these that we see is Down syndrome, followed by a cleft lip or a cleft palate. 90% of Down syndrome cases are due to an extra 24th chromosome. Chromosomal abnormalities are typically due to errors in cell division leading to an extra or a missing chromosome. Sometimes a gene is dominant for an abnormal trait, such as Huntington's disease. Dominant defects are less likely to be lethal. Recessive defects are expressed only if the child is homozygous for the gene. Recessive defects tend to be lethal. Incomplete dominance is a pattern of inheritance in which a child receives two different alleles, resulting in a phenotype that is a partial expression of a trait. I took this from your textbook. It's table 3.1, and it's some of the genetic defects that you may see. It is certainly not an entire list. I would suggest that you take time to read through this. One of the things you will see is the risk and the incidence of these and how often they occur, as well as what can be done in some of the characteristic conditions. Some of the diseases people can live with, and others, like Tay-Sachs, are degenerative diseases in which the infant always dies, usually soon after birth, and it is a horrible life for them. Studying heredity in the environment is important. We need to know how much does heredity in the environment influence particular traits and determine the relative influence of each. That is the question of behavioral genetics. Heredity is an estimate of the contribution of heredity and the impact of environment to individual differences in a specific trait. One of the ways that we study this is through twin studies and adoption studies. We look at what makes siblings so different. Children may live in the same family, but their experiences are not always identical. Non-shared environmental effects are the unique environmental influences that influences each child differently, including things like parent and sibling interactions, and illnesses, accidents, and various experiences outside of the home. It really is essential that we understand the role of the environment on development. It is the environment that we actually have the ability to control. Let's spend a little time on prenatal development. During this period from conception to birth, which we refer to as gestation, the child undergoes development. The normal gestation period is between 37 and 41 weeks. Gestational age is usually dated from the first day of an expectant mother's last menstrual cycle. Remember that conception does not happen until about 2 weeks later. The prenatal development period is divided into 3 stages: germinal, which is fertilization to 2 weeks, embryonic, which is 2 weeks to 8 weeks, and fetal, which is 8 weeks to birth. There are 2 fundamental principles of development to remember. Development is cephalocaudal, meaning it proceeds from the head to the tail. The head, brain, and eyes develop first and are larger than the rest of the body. The body eventually catches up. Development is proximodistal, or near to fore, which means near to the center of the body. So the head and the trunk develop before the arms and the legs. I'll repeat this again, probably several times this semester. The germinal stage, the first stage, from conception to 2 weeks, is the germinal stage. Fertilization to 2 weeks. Within 36 hours after fertilization, the zygote begins to rapidly divide, becomes more complex, and is implanted in the wall of the uterus. The zygote travels down the fallopian tubes and enters the uterus as a blastocyst. Cells begin to cluster. It takes about 3 to 4 days to make the journey through the fallopian tubes and implant in the uterus. The cells divide into the ectoderm, which will become the outer layer of skin, nails, hair, teeth, sensory organs, and the nervous system. The endoderm, or the inner layers, become the digestive system, liver, pancreas, salivary glands, and respiratory system. The mesoderm is the middle layer, and will become the inner layer of skin, muscles, skeleton, and excretory and circulatory systems. The amniotic sac is filled with fluid that encases the embryo and protects it and gives it room to move. The placenta provides the embryo oxygen, nourishment, and waste removal. The placenta is connected to the umbilical cord. In the embryonic stage, our second stage, which is from 2 weeks to 8 weeks, it is characterized by rapid growth and development of major body systems and organs. This is called organogenesis. This is when the embryo is at most risk to destructive influences in the prenatal environment. Risk of spontaneous abortion or miscarriage is highest in this stage. Most miscarriages occur during the first trimester or the first 3 months. A miscarriage after 20 weeks is considered a stillbirth. This is the period that the embryo is most vulnerable to things that can go wrong in the prenatal environment. The estimate is that about 1 in 4 pregnancies result in spontaneous abortions, and it may be as much as 1 in 2. The fetal stage is our third stage, and it is from 8 weeks to birth and is characterized by increased differentiation of body parts and greatly enlarged body size. The fetus grows 20 times its weight. We do at this time high-frequency ultrasounds, and we do this to detect the outline of the fetus and its movement. The first bones begin to develop at around 8 weeks. They can move and kick, make a first hiccup, suck their thumbs. By 12 weeks of gestation, the fetus swallows and inhales some of the amniotic fluid. A fetus can feel pain, but is highly unlikely before the third trimester because the nerve pathways to the spinal cord are not sufficiently developed until 16 to 24 weeks. Before 24 weeks, facial expressions of pain are almost absent. By 34 weeks, the fetus remembers voices. There are environmental influences that can impact the fetus, and most of these are maternal factors. Almost everything that influences the mom, from her diet to her moods, also influence the babies. It is one of the reasons we care a lot about tetragens, which are environmental agents that can interfere with normal development. These can be things like viruses, drugs, radiation, nutrition and maternal weight, drugs, alcohol, nicotine and caffeine, maternal illness. It can even be the mom's age, whether or not she's anxious or stressed or depressed. It's believed that mild stress may actually be good for the baby and it increases development, but high stress may result in irritability, impulsivity, and behavioral disorders. And then there are also outside environmental hazards. Did you know that women need 300 to 500 extra calories a day and they gain 16 to 40 pounds during pregnancy? In the U.S., about 21% of women gain too little weight and 48% gain too much. If a woman gains too much weight, she may have to deliver the baby by cesarean section because she's gaining weight, it's gaining weight. She is also at increased risk of preeclampsia, gestational diabetes, and postpartum hemorrhage. Malnutrition is a global problem that may be worsened by poverty, conflict, and climate change. Approximately 830 million people suffer from calorie deficiency. Insufficient calories during pregnancy can result in low birth weight or lack of nutrients like folic acid or folate can result in anencephaly and spina bifida. The environmental influences of the paternal factors are also important. So it's not just what the mom brings to the baby, but what dad brings to the baby. Abnormal or poor quality sperm can result from lead exposure, marijuana or tobacco smoke, large amounts of alcohol or radiation, pesticides, high ozone levels, and advanced age. I have provided you two tables like this for you to take a look at. They are slightly different, but they help you understand the same thing. Now that you have a basic understanding of gestation and factor, I want to show you these two slides to make clear why it is important to understand the process of gestation. The slide shows the three phases of development: germinal, conception to 2 weeks, embryonic, 3 weeks to 8 weeks, and fetal, 9 weeks to birth. If you look at the picture, you will notice that the orange is the greatest time of sensitivity to tetragens, environmental agents that can interfere with normal development. These correspond to the time of development of things like the central nervous system, the eyes, ears, palate, or brain. Notice that the critical period for the CNS is 3 weeks to 6 weeks, and for the palate, it is 7 weeks to 9 weeks. But there are less sensitive times when the development can still be impacted. This may be important when you are educating pregnant women on how to take care of themselves while pregnant. I included a second picture that essentially shows the same thing, but in a different format. This is important to understand because everything a mom takes in makes its way to the uterus. One of the reasons we do the extensive studies on drugs before allowing them to be prescribed is because of the use of thalidomide in the 1960s. It caused stunted or missing limbs in over 1,000 babies. It made us very aware of the dangers of drugs during pregnancy. The general rule is to avoid all drugs unless absolutely necessary during pregnancy. Likewise, alcohol exposure is a common cause of mental retardation and the leading preventable cause of birth defects in the U.S. Fetal alcohol syndrome and other less severe alcohol-related conditions are estimated to occur in 1 in every 20 births in the United States. Monitoring prenatal development, then, is essential. Healthcare providers have tools to assess an unborn baby's progress and well-being. These are things like amniocentesis, chorionic villus sampling, embryoscopy and fetoscopy, maternal blood tests and ultrasounds, pre-implantation genetic diagnosis. It should be noted that the more invasive procedures, like amniocentesis, have a higher risk of miscarriage than non-invasive procedures like ultrasounds. We also need to always remember that there are disparities in prenatal care. You would think we would want to provide the highest quality care to all pregnant moms, but this just doesn't happen. In 2017, 6.3% of pregnant women received late or no prenatal care. There is an ethnic disparity in fetal and post-birth mortality, which may result from this. About 830 women die each day from childbirth-related complications, and 99% of those deaths occur in low-resource areas. We can do better. In the United States, prenatal care is widespread, but it lacks uniform national standards. The rates of low birth weight and premature birth continue to rise, possibly due to an increase in multiple births and an uneven distribution of prenatal care. After adjusting for risk factors, the chance of prenatal deaths are 3.4 times higher for black babies, 1.5 times higher for Hispanic babies, and 1.9 times higher for other babies of minority status than for whites. As always, I invite you to office hours. Please reach out to me if there's anything you don't understand or if you need a little extra assistance. Thank you.